Hi, everyone. Thank you for the invitation to this very exciting conference. Unfortunately, I cannot be in person, but I'm really glad that the organizers allowed me to send my presentation and be there even remotely. So I will present you some results and some lessons learned from the European Marine Omics Biodiversity Observation Network, or else EMO BON. But a few words first about marine biodiversity. Of course, you all know it, and this is one of the reasons why this conference, I think, was designed, let's say, in the first place. Marine biodiversity is a core component of the ocean system. It has a lot of services. It's connected to society in many, many ways. There are many legislations regarding protection and restoration of biodiversity, such as the UN Decade Ocean for Science for Sustainable Development, the European Green Deal, where there are specific mentions for biodiversity observation. However, the capacity for biological observation is not as standardized or harmonized as people might say, as for the rest of the ocean scientific disciplines. And the marine biodiversity data are often fragmented in their collection and hypothesis-driven, which there's nothing wrong with having studies that are hypothesis-driven, but it doesn't really answer to the long-term questions that we have, and it doesn't really allow for the future predictions that we might want to do. So all these aspects gave rise to EMO BON, the European Marine Omics Biodiversity Observation Network, which is the first coordinated genomic observatory in Europe, with 19 marine participating sites, 20 institutes, and 10 countries. You can see in the map the locations of the institutes and the countries that are participating in EMO BON. What are the characteristics of EMO BON? Well, as I mentioned before, it's a long term. The plan is to have minimum 5 years of data collection. It started in 2021 and is still ongoing. Also, the main characteristic is that we want to create biodiversity data that are comparable. So this is why we have harmonized methodologies from conception to data collection and to publication, all steps of the way. Another characteristic is, of course, that it's a framework for genomics observation, not only in Europe, but something that can be replicated elsewhere. All the procedures, all the data and metadata standards that we use, all the regulations and guidelines are available for everyone to take a look and to use in their own observatory. And of course, it contributes to the global biodiversity observation networks, such as Omics BON and OBON, which I'm sure most of you are already aware of. So what is happening in EMO BON? Well, first of all, we have the sample collection. We deal with three types of habitats: the water column, the soft substrates, and the hard substrates. For the water column, we sample every 2 months, and we target the microbial community, and this is sequenced with shotgun metagenomic sequencing. For the soft substrates, again, we have every 2 months sampling for the microbial community and metagenomics, but also we analyze biofauna and macrofauna. And this is done for samplings 2 or 3 times per year, where CO1 and 618S RNA metabarcoding are being used. For hard substrates, we use the autonomous reef monitoring structures, the ARMS units. They are deployed long term, so for 12 months. And after the deployment, they are retrieved and they are photographed, and the organisms are scraped from the plates, and they are subjected again to CO1 and 18S RNA metabarcoding. So what is happening regarding the data flow? Because as you can imagine, all this creates a lot of data. What is sequenced with shotgun metagenomic sequencing? First of all, sequencing is being done centralized in Genoscope, along with the DNA extraction to minimize biases. All the data afterwards follow a data management procedure that is, let's say, not... it's very, very detailed and very thorough, and a lot of it is taking place in the GitHub site of EMO BON. The raw data are submitted in ENA. They are analyzed also with a MetaEcoflow pipeline that was built for this purpose exactly. The outputs of the analysis are packaged in narrow crates, which is a format of it's a format that allows, let's say, the full provenance of data and metadata to be combined together. So this means that everyone would be able to know where this sample was taken from, which are the sequences that are associated to it, what are the analyses that it was subjected to, what are the outputs of this analysis, and all this is packaged in a way that this information is never lost and everyone can keep track and backtrack of what has happened where. One of the things that we want to do afterwards, it's still it hasn't been finalized, is to find a way for Magnify, which is the analysis platform of EMBL EBI, to ingest those arrow crates and have the results already available in their website. From there, they would go to GBIF, OBIS, and Embodiment Biology, and then the European Digital Twin of the Ocean, EDITO. For the metabarcoding, the procedure is more or less the same, but some steps are simpler because of the data, the data type. Data analysis in this case is conducted with metabarcode analysis pipelines such as PIMA. We have a platform that has been built by LifeWatch Eric for this purpose. And then the outputs are being converted into Darwin Core archives, and they are submitted to OBIS and Embodiment Biology, and then the European Digital Twin of the Ocean. The second part of it, the Darwin Core generator, the submission to OBIS, is something that we are dealing also for we are trying to do also for the metagenomics datasets, because as I told you, the ingestion from the arrow crates to Magnify is not operational. So we are finding another way to submit their datasets to OBIS so that interested people can have not only the raw data, but have also the analyzed data. So some numbers. Since the creation of EMO BON, we have collected more than 3,500 samples. Most of them are watercolor samples, as you can see on the left part of the screen. And then we have the soft-substrates and the hard-substrates. The right part of the screen are the numbers. The numbers are not up to date, so this is why they don't match. I'm sorry for this, but it was a bit difficult to collect all the numbers in time for the presentation. But you can see more or less the analogy of the samples and how they distribute and what kind of size of data we are talking about. Yeah, and I shouldn't forget to mention that for these samples, or for all the samples, we also have biobanks. There a second set of samples that is stored and is not processed, and it's waiting for either for EMO BON partners, EMPRC partners, or external users in the future to be extracted and processed and sequenced using other protocols, perhaps, other sequencing techniques. We believe that the biobanking that we are doing also will open a door, let's say, in the past, in a way that it's not always easy with the samples that are already analyzed. So regarding some advancements, we have the first data release and the first data paper, and we have the second data release again in the same ENA project accession number, and the second data paper that was published a few weeks ago. Regarding ARMS, the ARMS project, again, we have two data papers. We have an ENA project accession number where everything is available, all the raw data. We also have everything on there, very, very thoroughly explained on their GitHub repositories. As I mentioned before, we analyzed the metagenomics data with the MetaGoFlow pipeline, and I'm going to show you some results and what we have come up so far. So just a brief overview again. Here is the map of the sampling locations. For this particular presentation, I selected some samples from the stations that you see on the screen. Where there is green, there are water samples. Where there is orange, we have both water and sediment samples collected. I didn't mention before, but not every site has to collect all three samples from all the three habitats, of course. We don't want to impose this to people, and we want it to be as easy for them to follow the protocols and to sample as possible. So for some results so far, we see that the water samples show temporal and spatial differentiation. We see that there's a clear distinction on the left part of the screen. You see in the NMDS plot that there's a clear separation between the filter size, because for the water samples we collect two separate filter sizes, and those samples are quite distinct. In the lower fraction, where we're supposed to have only prokaryotes, we also have some eukaryotes, 30% are eukaryotes. And in the upper fraction, things are more or less mixed. We also have prokaryotes there and eukaryotes. We see that the incidence is generally higher in the South European Atlantic shelf, so in samples from Portugal and Spain, compared to the North Sea and North Norway and Finnmark and Celtic Seas. You can see it, and the South European Atlantic shelf is the last one, is the last box plot that you see on your screen, the fourth one. We also found out that about 70% of all the taxa are shared between all the ecoregions. But again, we see a higher uniqueness in the South European Atlantic shelf. Just bear in mind that this reflects also the highest number of observatories and samples in this region. So these numbers are not, let's say, standardized based on the number of observatories or the number of samples that were eventually retrieved. So as you can imagine, this contributes to those numbers that we are observing. We also found out that there are comparing our results with literature sources that deal with pathogens, and trying to see if we found those pathogens in our samples. We saw that indeed we have them, we can identify them, which is nice, not nice because they exist, but nice because we can use our techniques as a monitoring method regarding also this aspect of biodiversity, regarding also pathogens, which we know are included in legislation and directives, and this is quite important. So we see, for example, that the sites that are close to ports, such as this some location in Bilbao, in the Basque Country, we see that we see the highest number of pathogens, the highest abundance, and we also see similar pattern in sites that are located in estuaries and lagoons, which would be more or less expected. Also, since metagenomics don't give also the taxonomic occurrences, but we can also retrieve functional annotations and functional abundances, well, functional potential, because we are dealing with DNA and not RNA, but still we can get a glimpse into the world of our microbes and see what they could have been doing. Getting those functional data and trying again to plot them in an NMDS, we see that we have again a temporal and spatial clustering in our samples, similar as to what we have seen from the occurrences. So we see that they kind of follow one another, they kind of match. So just a few words of how we can go beyond marine observation and what we could do in the future. So we know that EMO BON was created for marine observation, and that was the main idea behind it. But this doesn't mean that it has to be just that. The data that we produce are massive. They are publicly available. They just undertake an embargo of 6 months just to give a head start in the consortium, but they are publicly available for everyone to use. So they can be used for bioprospecting. They can be used for in studies regarding food provision and aquaculture. They can be used in taxonomic and phylogenetic studies. The sky is the limit. We try to do our work as best as we can to harmonize everything, all the procedures, and be as fair as possible. so that we can contribute as best as possible for the world and for people that are that share the same values, let's say. So EMO BON would not be possible without contribution of the local partners or EMBSA partners and people that are actually collecting the samples and processing those samples. And as you can imagine, there are quite many. So I would like to thank them. And I would like to thank you for your attention. And if you have any questions or if you have any input, anything that you can discuss, you can always contact me or contact also my colleague Julia Santi, who is the coordinator of the EMO BON. You can find everything on our website. And yes, again, thank you for your attention.